Welcome to On the Edge of Breakthrough, where hope meets innovation in the fight against cancer.
Hi, I'm Dr. Monty Pal. I'm a medical oncologist at City of Hope, and I'm really excited to bring to you On the Edge of Breakthrough, Voices of Cancer Research. We're going to be bringing some of our best researchers to the table to discuss exciting findings from their laboratories, from their research programs, and really get some great perspectives on what their plans are for the future. Welcome, everyone. I'm delighted to welcome my next guest, Dr. Enrique Velazquez Villarreal, a member of the newly formed Department of Integrative Translational Sciences here at City of Hope. Really great to have you here, Enrique. Welcome to the show.
Thank you, Monty. I really appreciate the opportunity to be here, and I'm very happy to share with the audience about my research.
Gosh, I have to tell you, you have such an amazing background. And if you don't mind, I want to take everyone back to the very start. How did you get into the field of medicine? My understanding is you started actually at a very young age. Is that right?
Yeah, so definitely I was like around 15 years. So I was inspired by my mom that basically she was looking to understanding herbs and how it works, people's health. And that really inspired me to get in the medical field.
That fundamental background and nutritional supplements and so forth is something that informs a lot of my own research. And where did it sort of go from there? As a teenager, did you start getting more and more integrated? I understand you started medical school actually quite early at 17. Is that right?
Yeah, that's right. So I started at 17 years old. I migrated by myself from north part of Mexico to even northern, where I basically in the city of Monterrey, when I get in this very competitive medical program, when we start like around 200 individuals, 200 medical students in order to graduate and get into this medical program where we got a lot of experiences, specifically in surgery, because this was a free hospital for everybody, hospital for the community. And that really helped me a lot to get all this experience and helped me to get into this area. And I tried to migrate again to the north, to the United States. And that's something that really motivated me a lot.
And a lot of experiences in between, right? So you were mentioning to me ahead of this program today that you had several mentors who were actually in the field of oncology that sort of pushed you in that direction, but several of them were afflicted by the same diagnosis. Is that right?
Yeah, unfortunately, I was very, I will say, affected by, cancer indirectly. So my MD mentor, neurosurgeon in the University of New Mexico, basically died because of colorectal cancer. And then also my advisor, my mentor in my PhD program, and my mentor in my postdoc program with the same disease, and it definitely this inspired me to get in one line of research, colorectal cancer, and try to identify cures for people.
I mean, it's such a special thing, that mentor-mentee relationship. Tell me a little bit more about that. What types of projects were you engaged in with them? How did they sort of influence your career trajectory?
Yeah, so basically they were very technological. My PhD advisor was working in supercomputer centers, basically also to the government, big data, big warehouse repositories. So I got this opportunity to get in this area of big data, of artificial intelligence, of machine learning. Being a physician and computational scientist at the same time just gave me this opportunity to integrate the data. And this is what I do every time. And that's why I'm in the Department of Integrative Translational Science, where I integrate data and I think from there. So I'm doing this every day.
And I mentioned at the outset your real international background, starting in Mexico, but also significant experience in Spain. Is that right, during training?
That's correct. So I got a fellowship in my third year as a med school, and then had the opportunity to go to Valdebrán Hospital, the Department of Surgery, and get a lot of experience there. And then did a lot of community training around in Europe, Paris and Rome. And this definitely helped me to increase my ideas about to apply traditional medicine. I will say clinical medicine, so clinical, like taking a little bit apart the technology, because, when you go to the community, there's not a lot of resources most of the time. And then I had the opportunity to return to Mexico with this training, with these abilities. And then I still needed the technology to complement and go beyond.
That's so interesting. It's when you mentioned Valdebron, for instance, one of the pillars in oncology research, I would say in Europe, Gustave Roussy in Paris, all of these amazing centers out there. Research sort of takes on a little bit of a different angle, doesn't it? Because out there, because of the limited availability of many standard of care therapies, I often find that patients are actually more inclined to participate in research. Do you find the same? Is that your experience out there?
Yeah, so communities are a little bit different there. I think they are more unified at some point by creating centers of, I would say, like poly centers. It's like, for example, Barcelona, Valdebron, a cosmopolitan city, and you see people from different parts of Europe, Africa, and also, from South and East Asia. And this is the way where you see the participation of everybody to help the physician to find a cure. So they are basically very engaging. And that was one of my experience over there. Then when I returned to Mexico, I felt a little bit different because then the community was more about receiving from the physician,
Okay.
like more receptive. So, and here in America, is like I see both models, working at the same time, broadening the system.
That's so interesting. Tell me about your experience in Mexico.
Oh, definitely. Yeah. So when I graduated from the University of Nuevo León in Mexico. So I had the opportunity to have my social service, and this is 1 year after your med school, to apply what you know and help to the community. So I had the opportunity to go to the National Cancer Institute in Mexico and the National Cancer Institute of Neurology and Neurosurgery, Manuel Velazquez Juarez, where I had this great time with a lot of physicians, scientists, that is basically in the capital, in Mexico City, to look for huge amount of patients, at the same time. We had consultations like, for example, we started at 7 a.m. and finished at 12 a.m., it's working for sometimes 16 hours without stop because the amount of patients that need help was incredible. So, and this helped me a little bit to understand that we are humans and we have a lot of limitations and think that technology could help us here because the volume. And that's why I start to thinking about, I need to know how to use computers, use technology to also help people because, I cannot work 24/7.
And one thing that I really strive to do, I haven't been able to do it yet, is really sort of cross that boundary in terms of research and do some joint projects with the NCI Mexico, with centers outside of the US. Do you feel like your body of research is kind of conducive to crossing borders and doing some international work?
Oh, definitely. So I have collaboration with the National Cancer Institute, actually. So my lab have international collaboration with them. And we're trying to figure out the difference of people that stay there and people that are migrating here, or people that basically born here that are Hispanic Latinos, and they are experiencing similar diseases. In the case of colorectal cancer, we see very similar statistics. So giving us this idea about like next to the environmental factors, is there anything related with DNA? So that's something that is definitely ringing our ears because they have an amazing amount of enthusiasm because they can easily start projects with thousands of individuals per day because the amount of need that people are looking for these centers as a hope to keep going in their life. It's a society that really want to obtain or get advantage of the technology that is in the center in Mexico City. So I think collaborations with them are essential to understand this part of the genome that also is spread, if we want to look like that, across not just the US, but across the globe. Because if we see DNA, it's 3 billion letters. These letters are coming from many countries and talk about the human history. So migration from years and years. So try to understand the sections of the DNA. Try to understand this risk, these mutations from one population can help us to infer risk for many populations at a global level.
I love that. And that's actually kind of a great segue into your broader body of work. And I wanted to talk about your sort of transition to faculty years here in the US. You've really had sort of a rich history of training here, getting your master's and then PhD. Maybe take us through that journey and onto your first faculty position.
Definitely. So after I finished my training as physician in Valdebrán and in Mexico City and in Monterrey in Mexico, so I got a fellowship and a scholarship to go to Harvard Medical School. For me, was super exciting. And get into the Valdebrán, into Brigham and Women's Hospital in HMS, and get this experience, a real experience with patients, in the U.S. and in the Department of Surgery. So this helped me a lot to get into this area of surgery. I mean, I was very familiar with the Da Vinci Project, using the robotic surgery. My mentor was one of the leaders in this area. And then at some point, I felt that I need a break to understand a little bit more about numbers, about computers to make research. So I say, I will stop here a little bit and I will do one master and we'll return. So that's what I say. So I applied to different masters, and the best one for me was at University of Pittsburgh. That was next to the Pittsburgh Supercomputer Center.
Okay.
And you say, wow, Pittsburgh Supercomputer Center, Bill Gates Foundation, NASA Foundation, all these beautiful minds there. In Carnegie Mellon, they have this supercomputer center and number one in computational science. At that point, 2008, 9. So I decided to go there, and start my first master's, a master focusing in statistics. Because, I say, well, I can go to the operator room, make different procedures, but I feel I need more numbers, in my mind to understand numbers and to understand statistics. It's to make research, to help better to my patients. And I start to get this master, mostly in statistics, get into program 2 years. And then after that, I say, well, but I don't know if these numbers go a little bit up and the computer cannot handle, so how can I make a statistic? So that's why I start thinking about doing big data. Then I moved to big data and then start to get a lot of different big repositories. And I start working at a WHO collaborative center. And, I'm doing a lot of statistics and epidemiology in public health. And then after that, one of my mentors, that basically was my PhD mentor, told me, well, it's great what you are doing, but just thinking that in the future, everybody will have a whole genome sequencing. And I said, what is that? Well, basically, it's like 3 billion letters. And if you can do statistics with that, that's the future. So I started thinking about that and say, well, how can I deal with 3 billion letters? You need to be a computational scientist. Oh my God. Okay, so what can I do? Another master. Okay, so get into another master. And then I finished with master and basically in programming inside the Department of Epidemiology. So a Master of Science, Master of Public Health, and then certificate in Big Data. And then what's the next step? Well, you have already all the masters. What about applying to a PhD? And I said, oh my God, another 6 years. So I get into a PhD in the University of Pittsburgh, joined with the Pittsburgh Supercomputer Center in Human Genetics. And good thing there is, the University of Pittsburgh has the medical center, UPMC, and the Carnegie Mellon University has the computational science part, that is basically the Pittsburgh Supercomputer Center. So I joined this together and finished my PhD. So I say, done. So my statistics, computational science, my medical skills, and then this is molecular. The molecular part done.
So the PhD's done. You're talking to your parents. Are they saying to you, okay, no, let's get that job?
Let's get a job.
Okay. So
let's get a job. And I say, perfect. So I had a great opportunity at that point to apply for different jobs outside in the industry. And then my advisor told me, I haven't seen somebody doing a lot of things. You really want to have a freedom. You need to go to the academia, because you have done a lot of things. So from the first year in the med school to the last year in the PhD, apply for a postdoc because you need to integrate everything that you have done. It's a lot. So try to integrate in one postdoc. So I say, okay. Again, one postdoc. So, but not 6 years, it's few years as a postdoc. So I started a PhD postdoc in a Scripps Research Institute, Before that, I had the great opportunity to meet a mentor. For me, was a mentor in Harvard Medical School, there's a Department of Genetics, Dr. George Church, and helped me a lot to understand and to see how the integration of clinical genomic data could help the future of healthcare. And with the resources I had at the Pittsburgh Supercomputer Center, I had the opportunity to get this coaching and helped me a lot to develop my dissertation. That basically my dissertation was using 100 million electronic health records, most of them simulated, with DNA and RNA using 42,000 terabytes, just memory, because it was an exabyte level, the hard drive. So just memory, floating the data and make all these beautiful queries in precision medicine, like asking individuals with certain clinical, genomic, and social determinants of health, and all these technical details or fMRI or all these technologies together, and then find the best medication in breast cancer. So that's what I was focusing on, and use different techniques because I was in computational science.
Oh, cool. Okay. And then did that path take you over to USC next? Was that your next stop?
That took me to Scripps Research Institute first for 2 years.
Okay.
There I developed AI systems for basically for the Scripps and 2 companies. So that was racing from Scripps. So, and this was about predict people if they will have acute rejection or subacute clinical rejection. That usually you don't know if they have a subclinical. Subacute reaction is just impossible, isn't it? As if this is not clinic, so you cannot measure that. But we can use the genome and check the function of the genes that basically I'm predicting you that in few more activation or increasing the transcription will start the point of acute rejection clinically. Basically, that was my project, a list of genes, 50 genes, 200 genes, 350 genes that predict acute rejection of every single patient just by having a drop of blood. In order to do that, of course, we went to the clinic, to the surgery, get DNA and RNA from people.
Okay.
Before and after the transplantation, before and after the procedure, and then get blood, tissue, and find the difference in order to get this 50, 200, and 250 genes that predict the acute rejection after kidney transplantation.
And so this whole pathway and genomics, where did you go next with it?
Yeah, so I had a lot of ideas to interact with industries around, become kind of advisor, like, for example, for our neighbors, like synthetic genomics, that they was basically growing mini brains, and they were doing basically trying to input 2 immune system in the same animal model, so the human and the pig system, and it's basically in the news. So they got $50 million from United Therapeutics. And part of my area was a little bit about having a discussion about how you integrate 2 immune systems there. So I have this kind of starting a collaboration with them. I had the great opportunity to meet 2 beautiful minds, in USC, that basically gave me the opportunity to start my postdoc, my faculty position in USC, and it was my first year.
And who are those 2 beautiful minds? Tell us.
Dr. John Carton and Dr. David Craig, and they basically opened the door for me to start thinking about how can I integrate data, how can I develop new methods on precision medicine. Now having this background, as I mentioned, a little bit of computational science, prediction, artificial intelligence, machine learning at the industry level, quality, and then the clinical data, statistical part, the genomic data, and then the multi-omics. That basically was my main point. Now, with them, I learned a lot about multi-omics, the real multi-omics, and how can I put this together? How can I integrate this to create better systems? And that helped me a lot to write the first software to basically understand the DNA at single cell. So at that point was like my first Nature paper, and was like, wow, for me, amazing, as being a faculty of first year, and understand how I can take from a tissue 1 cell, put in a drop of oil, and get 3 billion letters, and then another cell next to this cell, another 3 billion letters, and another 3 billion letters, 10,000 times, 10,000 cells, to understand the evolution of the cancer, because the cells have a different evolution, a different adaptation. And that was amazing. This is something that blew my mind, because now I say, this is my area. Now I'm doing clinical data. I'm doing genomic data using big data that suppose that I'm I'm doing that. And I use all my expertise in big data to organize this data, to write the software to organize all this knowledge and translate it to science, to discoveries. And for that, we use a colo-829, a very standard cell line. So basically was a very interesting discoveries.
Tell me about that. So with single cell data, this is something I always struggle to wrap my head around a little bit. You really do sort of capture tumor evolution. As a clinician, how do you think we can sort of utilize those findings in terms of colorectal cancer and its transition from early to late stage?
Yeah, that's an amazing question. And I'm still asking this question every single time. So when I see, cancer, I see an adaptation of cells. Now, for us, and basically in my lab, data is the heart of the discovery, and heart is dynamic. So every single pulse gives us this information that we can translate to cancer discovery and a potential cure for cancer. So we are using very dynamic systems to understand these cells that are dynamic. So when I see a single cell and I try to get this DNA, I say, this cell was from the past because it was an adaptation in an environment. The environment is changing every single time. And the DNA and the RNA is basically working and changing. So we see every single line at this point. And in order to understand cancer evolution, it's just to see these 3 billion letters of every single cell, how they interact with each other, and how can we find the subpopulation of cells that are susceptible for drugs. Mm-hmm. And then if you figure that out, so basically you can create the best medication for a single patient based on the evolution and the mutation of a group of cells that have similar targets or similar risks.
So you use this single cell RNA data, you identify these sort of unique subpopulations of cells, and you sort of come up with a therapeutic strategy based on that, right?
That's correct.
And you hinted on this colorectal cancer cell line, but I think what you're doing also is far broader in the context of patient-related data, right? I know from discussions we've had previously that one of your big areas of focus is this really terrible phenomenon that we're seeing in the clinics of young onset colorectal cancer. I think this is devastating. It's really an epidemic, isn't it? Any thoughts on what's driving it?
Yeah, when I see patients in their 20s, 30s dying because cancer, if you go to the registry, see your data, and then look all this mortality and incidence and prevalence in community in our catchment area, City of Hope catchment area, just around here. So we have an increased representation of Hispanic Latinos, and when we just try to subset the population, they are experiencing an epidemic. They are dying because this disease, early onset colorectal cancer, going up compared to other populations. So that is something that we really want to help, and that's what we are using all the technology that we have available to first understand what is happening. Is this the same cancer? Is the same biology? The other population? Is this the colorectal cancer as we know genomically, or is a different disease at the biological level? Is there the same genes starting the cancer as other populations, or are different genes? And that's what we are working very hard by using multi-omics, microbiome, and spatial transcriptomics, and all this technology to understand and then identify targets that can help us for drug development.
Well, what's your most educated suggestion at this point? So many people are asking out there, why are young people getting diagnosed with colon cancer? You outlined a bunch of potential factors, diet and lifestyle, perhaps. Certainly it could be a figment of underlying genetics, right? If you had to put your finger on it, is it one of those? Is it a combination? What's really at the root cause here?
Yeah, that's an amazing question. And I think a Nobel Prize will solve that question. And right now, what we have seen is we are trying to identify patterns. And the patterns indicates right now that this probably is a combination of genomic and environmental factors. And what we are doing right now is, as a comprehensive cancer care, we are looking for clinical data in our patients, the genomic data, and we are translating the social determinants of health. Because if you see the population and try to study social determinants of health, usually people in communities that live together have similar patterns in their activities because the zip codes, sometimes similar diets because restaurants are around, sometimes similar activities too because I will say the relationship between people, and we will say sometimes similar socio-economical factors because they live in sectors. All this help us to try to understand if that could also related with similar types of cancer. So, of course, we cannot answer the question right now, but genomics is giving us the idea that probably it's not that part. It's probably the genomic area. It's probably beyond that. It's having different genes coming from different mutations, founder mutations from years and years ago that are converging to the current population or the current generation. So I'm feeling it's an interaction, an adaptation, that we don't know and we need to search more.
And this is maybe another million, or billion, or even $1 trillion question, right? But how granular can we get with this data? We mentioned the changes in diet and exercise. We mentioned maybe somebody's underlying genetics, right? Which, ostensibly they might not be able to change. What about the intersect of that? I mean, do you think like diet soft drinks, for instance, particulate matter in the air? One of our thoracic surgeons, Loretta Ernsi, is a real specialist in that area and looking at alterations in one's underlying genetics based on air pollution. And could all these things be causing some evolution in our underlying genetic profile?
Yeah, you're totally right. I'm feeling this is something like an adaptation. Now we are ourselves. If we talk about pluripotential cells right now from the beginning of generating or growing a human body are basically experience interaction with different chemicals. So like soft drinks, you say was one example, but you see a lot of drinks that usually, we drink, we were, young, and then after that we check the ingredients are totally different. You can say, I don't want to say brands, but, just check, for example, this drink that you use when you go to the gym, and then, before and after, and before was salt and, 2-3 ingredients. Now it's like tons of ingredients and you don't know. I mean, you can mentally pronunciate them.
Yeah.
So I think the cells are right now trying to adapt to a different environment with a lot of chemicals that we don't know. And it's hard to test at a clinical level. We know how hard it is to identify if one chemical is basically related with a specific disease. This takes a lot of years. And I think the introduction of new chemicals right now is basically is so fast. And you see 3-4 chemicals if you follow the FDA regulations. The acceptation is so hard, so fast, and we cannot process that, and that's why sometimes we're thinking about probably we need an intelligent system to help us, to keep the same, speed.
My favorite subject when I was in college was organic chemistry, right? And, I have to tell you, I loved piecing together chemical structures. And when you look at the structure of things like aspartame and so forth, and if you know that sort of basic chemistry, you can see how they have this mutagenic potential, right? This ability to maybe take your DNA bases and mess with them a little bit, right? So I think the body of work you're doing is so key because you might be able to take that next step and say link potentially the use of these agents to some sense of causality in terms of whether or not they're driving colorectal cancer rates. I've got to ask you how you think that City of Hope and the research infrastructure that you have here is unique and potentially addressing that problem.
Yeah, so right now we have a amazing technology, I will say top of the line technology, where we can see the cells and beyond the cells, how they interact with other cells, and then take the DNA and at gene level, and even at nucleotide level, I mean letters, this 3 billion letters, so letter by letter, how they interact in one cell and then interact with another cell, and then they create an environment like one cancer cell interact with an immune cell, and then the cancer cell trying to protect itself, generate a necrotic environment, so to push out the immune cell and then adapt to a new environment where other cells cannot get into and get an approach and eat that cell. So we have that technology right now. This, of course, we are talking about cells. Just imagine the organs and systems. But however, with this technology we called spatial transcriptomics, we have the opportunity to obtain the data and find and check how patterns of cells, how they adapt to avoid like the immune system. And then how cells with similar mutations group by themselves, subsetting like a war, and then we see how they move around the bloodstream, for example, how they basically expand around the tissue. So the interaction with not just the cancer cells and the immune cells, but also the stroma cells that play a key role in this interaction, So you can see right now and then identify mutations, identify patterns that now you can basically look for different molecules that you can use to target that cells at the genomic level. Now, this is a spatial transcriptomics at the transcriptome level. Now we have another technology, spatial proteomics. Now, after the transcription, the translation of a protein. So we can see right now what is happening once the DNA has an affectation. How can we target? Because now we can target proteins, isn't it? Where they are. So that's a beautiful part. And I think this is pretty new. And we'll see a lot of this new research out. And beyond that, we have the multi-omics data that is basically not just the spatial transcriptomics and spatial proteomics, but we also have the microbiome, how this balance of microbes interact with these cells to increase the inflammation process, interact with the immune cells from the same patient. And this is giving us a lot of ideas.
Yeah, it's so funny how small the world is. Earlier during our conversation, you mentioned David Craig, who's the head of your department, John Carpton, who's now, of course, at the head of our cancer center, just amazing individuals. And, Dr. Carpton, as I understand it, has his roots in TGen, which happens to be a City of Hope subsidiary in Phoenix, Arizona. And so they come to City of Hope, just like yourself, with tons of incredible experience, as it pertains to genomics. And it's lovely to see all these different worlds meld, because I've seen some of the evolutions of these different genomic spaces independently. The microbiome, spatial proteomics, tying them together. I mean, it's so incredibly complex. I'm glad we have folks like you who can really integrate very nicely. And I think that's going to be key for our development here.
Oh, thank you. Yeah, it's what we do every day. Yeah. So, and we are using the top technology. We develop the technology too. So we are at the top of generating like intelligent systems to help us to integrate this data.
So that's actually the next thing I wanted to talk about, because whenever we talk about big data, I think clinicians like myself, we get a little nervous, right? I mean, you can be so eager and enticed to look at, for instance, a gigantic data set, looking at the microbiome or perhaps a patient's genetics, but you take those different data sets, which have millions, if not billions, if not trillions of data elements, and try to merge them together. And sometimes you end up with a bit of a huge mess, right? And what's dangerous, I think, is for somebody like myself to dabble in it. But what you've created is this amazing tool, and tell me if I'm describing this correctly, but it uses artificial intelligence. So you can take I'll call myself a genomic simpleton, okay? Mm-hmm. And I can take, for instance, the population of patients that I'm interested in, kidney cancer, and just say, can you dissect out, using your AI tool, a genomic difference between population A and population B? Maybe it's younger patients versus older patients. Maybe it's Hispanic patients versus non-Hispanic patients. And it essentially spits out all of the relevant genomic information that really dichotomizes those populations. Am I on the right track with this?
You are totally right. Yeah. So, and this is an AI system based on agents. So we call the AI agent system where basically our conversational tools. It's a chat, So you can even input voice recognition, so you can use your voice and you can type. So these systems are based on a cognitive model that we basically developed from scratch, 1 and 0s. And this model was trained to decide between A and B. What is A for me? What is B for me? And it's basically thinking about, understanding about what the difference between A and B, and you say two groups, isn't it? So these cognitive models that were developed when I start here at City of Hope start growing, in recognition of patterns. And then they were developed and basically trained to identify case control studies. So these are like babies, so you have a system, you put the cognitive model and then start to asking the model, A is less than 40 years, B is more than 40 years. So now this is a database dividing A and B. Sometimes it's doing right, sometimes not. It's a cognitive model, it's not a pipeline. And then you teach again. Okay, so you got 40 inclusive. Now, 40 is not inclusive, 41 is inclusive, and you start to polishing the model. Day by day, pushing data, and then the model is recognizing and is giving you patterns until it's working like with 100% accuracy, that you can measure that. So, and at some point, this model, and we are pushing out as diversity in AI development, because it's basically what we are pushing out is a new model, different than GPT chat. This is a new GPT chat for the lab, and having a interface that we generate. So we basically are trying to help us, because this is an assistant. It's helping us to understand and deconvolute this data. Now, the great point is, yes, you can divide in case controls, and these agents are not just divided in two, but also are studying one by one, are getting into the facts, are going to the literature, and getting results and analysis for you in a couple of minutes with a PDF sending to you to your email, and you are getting all the analysis based on your query. So basically, it's an assistant, and also it's asking you for the next step. So that's what we are doing to deconvolute this kind of data.
I love that. And by no means would this sort of substitute bioinformaticians, right? I mean, there's still such a huge role for that. But with that in mind, I think what this does as a clinician is it allows me to at least take that first step and ask a question, right? And then maybe go to that bioinformatician and say, can we dive a little bit deeper here, right? I mean, I see real value in that. One of the things I wanted to emphasize about what you just touched on is that this is homegrown, right? And that's so key, I think, because, we've all got a chat GPT. I've done this for kidney cancer, just to provide a quick update, for instance, on the latest and greatest in therapy. And it confabulates, right? It does that on occasion. It'll pull resources that I would say are 90% valid, and then it kind of makes up the rest, right? And there's room for that, I suppose, in some exercises, but certainly no room for that in looking at big data. Am I right?
Totally right. Yeah. So especially because, these are systems that are local AI. We know where the system is. We are basically, we have this portability. Everything that we do is inside a specific computer, and we know that is closed system. So that's good. It's private data. And what you say about other big, I will say we have just GPT chat and DeepSeek, and a few models there, but not a big arsenal of these options. So when you go to these models have restrictions. There's some point where you cannot go beyond certain language. So, and we're designed for populations, not in science, for populations directions, so to help the population, to identify queries and to answer questions for a population. So our software or agents are free, no restrictions. Focus on clinical data, genomic data, nature papers, science papers, and inside an environment of science developed by scientists with different thinkings that social media and the community computational science scientific population. So we are basically generating this with one specific target: find cure for cancer. And this is what our models do 24/7. Identify targets, check overall survival, should be the best for the patient. Now, we are including clinical, genomic, and social determinants of health data. And what I'm doing is, because this system, or systems, because I can say, are free to everybody I use public data so people can test this and can trust on the system. Beyond just putting a wall, private data, and nobody can use it, who will trust on this? So I'm trying to, to open the AI, without talking about the company, it's just like to people understand that these models can help and everybody can build their own model to help their research. And these models can help you to analyze data. So in my lab, for example, and I have several videos in LinkedIn when my staff are basically doing their job in bioinformatics, analyzing the data, RNA-seq, DNA-seq analysis, and beautiful clinical genomic data integration, models. And then on the side, we have our basically interface with AI Hope, that is how we call it. So, and then can you do this analysis? 2 minutes versus my staff 2-3 weeks. And they go directly to analyze all the mutations and everything. And we are comparing because we don't trust on the system 100%. We need time, and we are improving this every single time because this is not a pipeline when you say, oh, here is the problem. So I need to modify this program to keep going with the flow of the data analysis. This is a cognitive system. It's an intelligent system. It's taking prompts.
That's so cool. And it really does seem like you've come full circle, right? We opened today talking about your background working in Mexico, starting those rotations at the NCI Mexico at 7 a.m, working till midnight. That's certainly one way to contribute to the field, working around the clock. Now we've got computers doing that alongside us. And that's a super, super powerful tool. Now, a lot of the research that you've done has culminated in what I would say is a really outstanding and prestigious award. And you strike me as being a really humble person, but I'm going to ask you to brag for just a second. Okay, you can do a humble brag, as I like to call it, right? But tell us about this AACR award that you're getting.
Yeah, so we have the opportunity to show our work to, submit our work to AACR. This is basically 4 projects. So one project is the AI, an agent. And I think for first time in AACR will be what agent participating in showing how you can integrate clinical genomic and social determinants of health together. And this is one project. The second project is about spatial transcriptomics and how we integrate this data with clinical genomic data. And the other project is a colorectal cancer disparities in Hispanic Latino population. And for first time, we will have a big sample size for this population. Probably will be for first time in history having all this amount of Hispanic Latino clinical genomic RNA and DNA data together and showing the frequency of the mutation of colorectal cancer of early onset and late onset. And the last project was about proteomics analysis. I think this technology is pretty new, so there's a few projects are there. And to explain how new software can help us to analyze this proteomic data and translate this to the clinic. That is our main point as physicians. We want to translate it to help us, to help our patients. And I think AACR look for this work, and I'm very honored to get this award based on this work because it's focusing on minorities population, underrepresented population. That part of my lab is generating DNA and RNA from this population to public data to push research out of these individuals, especially in the Hispanic, Latino, around the LA area, inside the LA area, and around the LA area.
What a great way to contribute. I mean, gosh, I really see so many roots of this in your heritage coming back to sort of serve the Hispanic Latino population. It's just remarkable. I took on a role as vice chair of academic affairs of my department of medical oncology about 3 years ago now. And it's probably been one of the most rewarding things that I've done. And one of the things I like to do is sit down with folks that have just joined City of Hope and just ask them this question: where do you see yourself in 10 years? Now, you've already done a lot more than many of our junior faculty at City of Hope, the multiple postdocs, the master's degrees, et cetera. Where do you hope to see your program?
Yeah, so in 10 years, I visualize myself basically with automated AI doing a lot of DNA and RNA integration for clinicians just before going to a surgical room. So it's before going to the clinic, and summarize them in a simple way, in a practical way, how they can better treat every single patient at individual level. So that's what I'm trying to do. I'm pushing a lot. It's a huge step, but I'm feeling like I visualize myself, providing the systems, that help people in the front line as you working hard, to beat cancer.
I love that. I think one of the main tenets of success here at City of Hope is just having that underlying clinical goal, right? It can't be to publish in Nature or Science or what have you. Those are laudable goals too, but separate. I think if you're really driven by this goal of helping patients, that's really when you're going to succeed, right?
Definitely. I mean, this is the translational part. I think this is my work. When I see research and I go to all these, amazing meetings, scientific meetings, I always think about, part of me is, okay, so translate it. You are the translator, translate this to the clinical part. If I have a patient tomorrow, how can I help with what I'm listening right now? And that's why I also apply, Arsena, one of our AI agents for clinical and genomic data integration, to ask for first time, and was accepted just yesterday.
Okay,
so we got another AI agent there to explain the scientific and the medical community how it works, how this could help you to develop new tools by generating your own cognitive model. Because I think everybody are brilliant, can basically develop their own models and help themselves and help people, and everybody, together, we can beat cancer by putting all the knowledge and all the different, thinkings and different point of view, and build different systems that at the end of the day is one system, to help to understand the data, to understand the data's patient, to understand the DNA at the end of the day, and once you understand the software of life, you definitely will win the battle.
Brilliant. Very well said. Yeah, I saved one of my toughest questions for last here. The title of this program includes On the Edge of Breakthrough, right? So what does that mean to you? I've asked this question to everyone sitting in that seat, and I've always been impressed with the answer. On the edge of breakthrough.
So mean translational and AI to fight cancer and beat the fight.
I love that. So taking current tools, right, really applying them, all the amazing diagnostics and everything that we have in computational biology, and using that to sort of redirect our efforts in cancer care.
Yeah, definitely. We are in 2025. For me, this is, it's a part of the AI revolution. It's part of the start of everybody writing code, and to everybody want to keep going in these with this revolution. And I'm feeling like the cancer research need to adopt this revolution. And for that is like when, the digital era start, everybody changed from one type of phone to digital phones, so it's the same way. So we are just translating what we do in our computers to cognitive models and doing beyond what we can do as humans. And I think that's something that in cancer research I want to basically, spread the word. Use AI to increase your abilities, to increase your opportunities to succeed, and generate your own AI.
So to summarize, DIY, do it yourself.
Yeah.
Well, I really enjoyed our conversation today. This has been absolutely terrific. And I think this 10-year program, this 10-year plan that you've established for yourself, I think you're going to realize it. I think it's going to happen here.
We're trying every day very hard and with a lot of passion. And and of course, we are having, this commitment. Every time I come to City of Hope, I try to walk around, the patient area, they inspire me, they give me this energy that I need sometimes, especially very early in the morning, to get this, increase my commitment and get clear ideas about how can we change that, and I remember the podcast with Kim is to think out of the box, so, and try to be, or try to move to this innovative area, to help people in a different way. You lead the work to cure cancer. And that is basically what I'm trying to do every time, just get this beautiful thinkings from patients, when I go around and just look them, just hear the conversations, sometimes I'm having interactions with them. And this really fit me as a scientist to find a way to change the things that basically are normal for us, your cancer patients. How can we change this world to non-cancer world?
Beautifully said. On that note, thank you so much for joining us today.
Thank you so much.
This podcast was produced in partnership with Amaze Media Labs.